Hello, I'm Dr. Anna Pyayt. I'm head of the Innovative Biomedical Instruments and Systems Lab. I'm a faculty member from the Department of Chemical and Biomedical Engineering, College of Engineering at USF. And first, I'm going to describe my background. I did my dual PhD in electrical engineering and nanotechnology at the University of Washington in Seattle. And during my time at the PhD program, I was intern at HP Labs at Microsoft Research. It was a very interesting and important experience. At Microsoft Research, I was able to work with Gary Starkweather, a laser printer inventor, and we had a wonderful publication about telescopic pixel new display technology published in Nature Photonics. After that, I received a 2-year Microsoft fellowship that allowed me to finish my PhD. After that, I received Computer Innovation Fellowship, and this Computer Innovation Fellowship allowed me to do my postdoc at Stanford University, which was another amazing opportunity. And after finishing my postdoc at Stanford University, I started my position at the University of South Florida, and I'm currently an associate professor at the Department of Chemical and Biomedical Engineering. Here you can see my team. I work with different students from undergraduate to graduate level. Some of them are master students, some of them are PhD students. They have different backgrounds, they have different expertise. Some of them are working with cells and bacteria, while others can do computer programming. Some of them can work with Arduino, and others enjoy 3D printing. So we need a really wide variety of skills because we are trying to create fully functional instruments or systems. I briefly describe several of my projects and show one of them in a little bit more details. So I'm just going to show you one slide for telescopic pixel, for single cell endoscopes, briefly talk about Hemolex and mobile ELISA projects, and spend the rest of the time describing optofluidic particle manipulation. The telescopic pixel project is the project that I did in collaboration with Microsoft Research. And we have a patent on that and a publication in Nature Photonics. In addition to that, the original publication was translated in 32 languages and it appeared in all major news outlets and so on. And the reason for that is because a new display technology that we proposed was 5 to 10 times more energy efficient than traditional LCD displays. The Hemolex technology is a different type of technology I have been working on while being at USF. This is mobile phone-based technology that allows us to detect different pregnancy complications. With this technology, we were able to get into final of X Prize competition. We were finalists of Vodafone competition. We presented this technology at several major places such as US Congress and National Academy of Sciences. So overall, it was a very successful project. Another mobile phone-based technology that my lab is working on is called mobile ELISA. And it's a very powerful platform that allows to conduct traditional ELISA tests using very small instrument shown here on the screen. And ELISA tests can be used to analyze thousands of different proteins, biomarkers, hormones, and other important molecules. And our lab was the first one who figured out how to scale down two huge instruments traditionally used in labs into one small device and a mobile phone, and we can run full functionality of ELISA using this small low-cost instrument. So this project was also in the news. Elsevier chose this publication among thousands of other publications for international news coverage. And we also received support from Amazon and from internal USF grant, because this technology can be used to analyze COVID-19 related antibodies and understand immunity for COVID-19, which is extremely important right now. Another type of technology my lab has been working on is called single cell endoscope. This is a very different type of technology, again, very interesting and innovative. And it's based on extremely sharp tips that can be inserted inside of individual cells and cell organelles and analyze the structure of the cell. The tips are so sharp that the very tip can be down to 50 or even 5 nanometers in thickness. And if you think about traditional cells that are used in the lab, they can be around 10, 20 micrometers in size. So tips that are so sharp can be inserted inside of those cells in individual cell organelles, and they can pick up optical signal, and the signal can be used for analysis of the cell. We collaborate on this project with a really large number of different researchers. Some of them are from Moffitt Cancer Research Institute, so we analyze different types of cancer cells. We collaborate with Public Health, College of Public Health from USF, and we were able, for example, to scan nucleus of individual liver cells, and we showed that DNA is distributed non-uniformly throughout the nucleus. And you cannot just obtain this information using traditional microscopy, because different layers of nucleus would conceal this information. It's not transparent, completely transparent when it's labeled with fluorescent dyes, while our technology allows to pick up signal very locally, and as a result, we can get very detailed information about structure of individual cells and cell organelles, which is very interesting and very exciting. And the project that I'm going to discuss in a little bit more details is our technology that is based on optofluidics. And you can see that when we work on a project, we need to take into consideration many different aspects. We need to run experiments, we need to run simulations, we need to do optimization of optical parameters, and we need to do optimization of other physical parameters. We need to use different software and so on. And each individual aspect of this research can be done by a different person. Because of that, it's definitely team effort. And when we all work together, we can actually make something very interesting. So here, I start with COMSOL simulation of simple optofluidic configuration. So imagine that you have cylinder filled with fluid, and at the bottom surface in the very center, you are heating the surface. Okay. So the heated fluid is going to have lower density and it's going to go up. And as a result, in this little well, you are going to establish current that is going to have toroidal shape. And in order to be able to optimize all these parameters and understand the temperature distribution and fluid movement and all these things, we actually use COMSOL multiphysics simulations, and different aspects of physics are described with different equations that are shown on the right. Okay. So when we understand what kind of current we can establish in this configuration, the next question is how to make this kind of structure and what are potential applications. So when we start with design optimization, we need to optimize for many different parameters. For example, we need to figure out what is going to be this bottom surface that can be heated. What are the heating mechanisms? How to optimize it? So we choose light for heating the surface, and because of that, we need to create some kind of substrate that is going to absorb light, and it's going to generate this toroidal current very efficiently. There are many different structures that can be patterned on this substrate, but based on simultaneous optimization of optical and thermal properties, we figured out that we need to create continuous metallic bilayers. And one metal is going to be chromium, while other metal is going to be gold. And the reason for that is because we want to create something that is biocompatible, that can absorb light very efficiently, but at the same time is going to have really good thermal conductivity. So we would be able to generate these currents very efficiently. This required a lot of optimization. Part A of this figure is optimized using COMSOL. Part B is optimized using specialized optical software. Part C shows our measurements that were done using thermal camera, because we were able to fabricate this substrate and actually test its performance to make sure that, yes, indeed, we can very locally heat the substrate and generate those toroidal currents. And part D shows our testing, and the temperature of the substrate was linearly increasing with the power, and this results in faster and faster moving currents. Okay. So if you look in this image, it shows you the schematic of our optofluidic setup. We have a laser and optical fiber delivers light to our specially optimized substrate. Everything is observed using microscope lens. The experiments are conducted in special microfluidic chips, and in simplest configuration, we can have two different regimes. When we're using lower light power, we create this toroidal current, and this current can bring particles into the center of the structure. However, if we increase the laser power, we can actually levitate the particles that we brought to the center. So the first regime we call the trapping regime, and the second regime we call the levitation regime. This is how the whole setup actually looks in real life. And as I mentioned, we need to incorporate the structure into a different type of small microfluidic chips. So we first create our substrates, and then we use PDMS and either PDMS wells or channels and integrate these structures into these microfluidic channels. You can see how optofluidics can be actually used to capture beads, which is shown on the left, or capture and levitate beads. You can see how they are captured on the right. And part C and D show little streams of particles that are projected upwards. In addition to that, we found out that we can use this approach to selectively project and capture two different types of particles. So if initially we have a mixture of larger beads and smaller beads, we can use optofluidics to capture them in the same area, but after that we only project the small ones, and as a result we can selectively collect all large beads in one place and get rid of the small ones. The video on the left shows how we use optofluidics to collect the particles. And the video on the right actually shows you projection. So it's done at angle, and you can see those little particles flying away. In addition to that, we tried the same technology with a variety of different cells, microscale objects, and even live organisms. On the left, you can see C. elegans worms that were captured using this technology. And this is the largest ever live organism that was optically captured. What is also very important is that the worms were completely fine. They were not damaged. They were feeling great after we captured them using this approach. So there was no thermal or optical or any other damage induced on those worms. And to demonstrate that this technology can work for a variety of objects, we captured silver nanowires, and you can see scanning electron microscope images of meshes of those nanowires. And the nanowires are tiny. They are measured in nanometers range, while live worms are 1 millimeter in length and 50 micrometers in size. As a result, those two types of objects have 7 orders of magnitude difference in mass. So this is a wonderful dynamic range of this technology. And in addition to that, we studied many different configurations of this technology. We incorporated it into tiny microfluidic channels and studied how particles are captured when external flow of the fluid is present. We did a lot of additional simulations using COMSOL and looked at how this toroidal shape of the current is disturbed in presence of external flows. We studied different conditions for which current changes, and at the end it completely transforms and it cannot capture any particles anymore. We looked experimentally what happens when we interfere with the toroidal current and introduce additional flows into microfluidic channels. And Here you can actually see how particles are captured, and when additional flow is introduced, the whole cluster of particles is shifted relative to the initial configuration. So in addition to just capturing and filtering particles, we wanted to make sure that we can isolate needed type of particles from the microfluidic chip. So we introduced tiny, very, very thin needles into our microfluidic chips. And you can see how particles are captured, and then we use the needle to capture the trapped particles. We did many studies showing that our chips are biocompatible. We were growing cells after manipulation with optofluidics, and we found out that cells were growing just fine. We don't damage cells, we don't damage the whole worms. So it's a very safe, gentle micromanipulation technology. We had many experiments with live bacteria and also demonstrated that we do not damage bacteria this way. We were able to successfully concentrate bacteria and isolate them from biological samples. And what you can see here is actually little bacterial clouds that we were able to capture from a large biological sample. In addition to that, we collaborated with Professor Suarez lab, and we can use this bacteria to prescribe beta antibiotics. So the whole idea behind this bacterial project is that sometimes patients has to be treated against bacterial infection, and in some cases, for example, when it's sepsis or a really complicated bacterial infection, the antibiotic prescription cannot be done in a correct way because all the traditional tests take several days. So we wanted to speed up this process, found a way to isolate live bacteria from patient sample, and then choose the most efficient antibiotic. So here I want to end my presentation. You can go to my webpage, www.pyayt.com, if you want to learn more about any of my projects or read my publications. There are more interesting projects available there, and I'm always open to work with new students. Have a nice